Hello friends, now in this lecture we are continuing with Meiosis II. Now in Meiosis I we have divided a diploid cells into two haploid daughter cells. So right now into Meiosis II we have two daughter cells, one and two daughter cells we are having. But thing is that it is having haploid condition with doubled chromosomes. So chromatids are into the pair in the form of chromosome into the haploid condition. They are same homologous chromosome. At one side we have maternal, then in the other side we have paternal chromosomes. Now these chromatids has to be separated and divided and will have to send into separate cells to produce haploid gamete. Which should not contain double the chromatid but a single chromatid. So single haploid gamete, single in the form of or in with respect to chromatids, single chromatid containing haploid gametes should be formed during meiosis II. So in this phase, entire mechanism of separation of chromosomes is happening into the similar four phases whatsoever we have seen into the mitosis: prophase, metaphase, anaphase, telophase, but it is into the meiosis II. That's why prophase to metaphase to anaphase to and telophase II. Now let us definitely remember that meiosis II resemble mitosis. So there is no change at all into any of the phases into the mitosis or meiosis II. Both are similar. Ready? So if I, in the other word, what I need to say is I am revising the mitosis again. But only thing is that here in meiosis II it is happening with two doubly or doubled haploid cells together. So both the cells are going with a divisional state which is similar or resembling the mitosis. Ready? So that's the difference what we can see in this. So let me quickly revise the entire thing which is seen here. If you see, during prophase, the main or chief differences of the meiosis and mitosis, for example mitosis if we have discussed whatsoever, prophase, metaphase, anaphase and telophase. This was the first discussion of the prophase, metaphase, anaphase and telophase of the mitosis. So during prophase of the mitosis, right, this is the prophase what we have seen. So condensation of chromatin into chromosome, that was the main event what we have seen. Second change what we have observed during that is disappearance of nucleolus. Why? Because of chromosome appeared and chromatin disappeared. Third change what we have observed there is disappearance of nucleolus and nuclear envelope as well. So there were three changes. Now if I talk about right now condition, that is meiosis 2, two things are happening. Nucleolus disappears, nucleolus and nuclear membrane disappears, but chromosome condensation already there, so there is no condensation of chromosome, it is already there. So it is initiated immediately after cytokinesis of meiosis 1, and the chromosomes again become compact, that is nearby, arranged nearby, but there is no condensation of chromosome is seen during prophase 2 of meiosis 2. So that is how it is very similar, except condensation, because condensation is already there, it has not to happen here. Ready? So please take the screenshot. Now metaphase. If we remember the metaphase, what we have seen during mitosis, it is the chromosome in the cytoplasm. Now sets of chromosome, they are to be separated, arranged into a particular manner. Spindle fiber have been reformed, attaching to each chromosome at the centromere, and arrange them into the equatorial plate called mitotic plate or metaphase plate. So that metaphase plate formation is a key feature of the metaphase of mitosis, and that similar mechanism we can see here. If you see here, the chromosome align at the equator similarly, right? So chromosomes align into the equator. Now microtubules, that is spindle fibers from opposite poles, they get attached to the kinetochores of sister chromatid at both the sides, similar to the the case which is seen into the metaphase of mitosis. So again, at both these sides, microtubules are attached at the place of centromere on the surface of kinetochore. So that's the event which is seen into the metaphase 2. So mitotic or metaphase plate, single equatorial plate is again formed. Now if I talk about Anaphase 2, similar to that one, similar, right? So Anaphase, if we remember, separation of two sister chromosomes, that is chromatid of the chromosome, that is homologous chromosome, from centromere region. Pulling and pushing movement, what we have observed, what we have seen, what we have understood. So two sister chromatids are getting apart from the centromere region. Why? Because decreasing length of spindle fibers. Contraction of the spindle fibers happened during the anaphase. So similar mechanism again, this is metaphase, this is the spindle fibers, and that will be contracted towards their respective poles. That is why the chromosomes are detached, getting separated or splitting out from centromere region. So one chromosomes, this one, will be divided into two sister chromatids, will be separated towards their respective poles, right? So this way both the sister chromatids are moving, first splitting, then moving toward their respective poles, either north pole or south pole, and that is the end of the anaphase. So what we can see is it begins with simultaneous splitting of the chromosome, centromere of each chromosome, which was holding sister chromatids together. Thus, when it is splitting, then they move toward opposite pole of each cells. Easily they can be detached by splitting, and that is why, due to the contraction of the spindle fibers, they are moving towards their respective poles. So that's the important phenomena which is happening in Anaphase. Please take the screenshot. Now, telophase. Now, as we know that during telophase, very important thing what we have noticed during whatsoever we have understood in the mitosis is formation of two daughter nuclei, reconstruction or opposite work from the prophase. For example, chromosomes, they are separated back, decoiled and becomes chromatin material. Nucleolus reformed from the nuclear region, nuclear envelope which is actually encoding the entire chromatin will be reappeared, and spindle fibers, they will be diminished, they will be disappeared, so there will be no spindle fiber later on. So these are the necessary changes what we have observed during the mitosis telophase. Similar thing will happen here. So during anaphase, where the chromosomes are getting separated, they are actually combined, they are actually gathered towards their respective poles, and nuclear envelope will be reformed at both the sides, right? So nuclear envelope will be reformed, so two nucleus, then it will be following the cytokinesis as well, and two cells will be separated out from one. So the changes, if we want to observe during that, is two group of chromosomes once again get enclosed by nuclear envelope, then comes the cytokinesis resulting into formation of tetrad of cells into four haploid daughter cells, two of each cell, so one and the other, so total 1, 2, 3 and 4 daughter nuclei will be formed, and that is why 4 daughter cells will be formed out of 2 homologous pair of chromosomes containing single cell and the other cell. So 2 cells will be actually doing the same process of prophase 2, anaphase 2, metaphase 2 and telophase 2, and that is why 2 plus 2, 4 daughter cells, haploid daughter cells will be produced, that is gametes. So entire mechanism, if we conclude, please take the screenshot. So entire mechanism, if we conclude, see overall step, if we see prophase 1, metaphase 1, anaphase 1 and telophase 1, it is meiosis 1, right? And this is meiosis 2. So here one cell is there, here 2 cells are there. So 1 becomes 2 and 2 becomes 4. So 4 haploid type of gametes will be produced. If I actually mention the cell type, this is diploid cell, this is doubled haploid daughter cells, and these are single haploid daughter cells. Instead of daughter cell, I can definitely write the gametes. So this is the mechanism of production of the gametes. During gametogenesis, meiosis is the process which is happening. Why? Because we want to produce singly haploid gamete. We want gamete into the form of haploid condition, into the having a single set of chromosomes, not double set of chromosomes. Why? Because as we have discussed earlier at the starting of the chapter, that zygote, which is a starting point of our life cycle from where our entire body is formed, is a result of fusion of two gametes. Both the gametes, if are diploid, then zygote must be tetraploid, which is not possible. So in order to maintain diploid condition generation to generation, we need to have the gamete into the haploid condition, and for producing the gamete into the haploid condition we requires meiosis process. So that's the importance of the meiosis. Second importance of the meiosis is to produce variation. So by crossing over during prophase 1, what we have seen, crossing over and the exchange of genetic material, that is recombination. So crossing over and recombination is resulting into variation, and that's the importance, two main importance of the process of the meiosis, what we say. So please, you can take the screenshot of the overall process, overall steps of meiosis. Now, the significance or importance of meiosis. Now, based on the direction or based on the steps which are giving here into the form of the few cells diagrams, if you see, this is the maternal cell, right, diploid cell, what we can say, which is going through the process of meiosis. So during meiosis 1, they are exchanging the genetic material. If you see here and here into the chromosome, only single color is appeared. It is plain white, and this is yellow, red in the color. But if you see, here is the mixture. Why? Because of the crossing over, because of the recombination, genetic material of the chromosome is getting exchanged between the chromosome, and we know the process. We have gone through entire process of meiosis 1. So at the end of meiosis 1, two daughters nuclei containing cells are produced, which is having haploid but doubled chromosomes. So it is reductional division. Meiosis 1 is reductional division. From diploid, it is converted into haploid. And then during meiosis 2, we are separating them, these chromosome, homologous chromosome, into the respective two different daughter cells. So daughter nuclei number 2 will be containing single haploid cells, right? So that is how entire process works. So two important things or events which are happened here is the significance what we are getting from it. Conserves the chromosome number in each species. Here I must say generation by generation. So diploid generation will produce diploid offspring only. Why? Because of the process of division of content of chromosome during the meiosis phase. So two haploid gametes will be produced, will be fused and produce zygote, which will be containing similar or identical chromatic or number of chromosomes into the next generation as well without change. Now the second important phenomena what we are observing is it causes genetic variation due to the crossing over into the population of the organism. It is important for evolution. So new types of recombination will produce new kind of characters, right? So as and when any of the organism which is actually reproducing by sexual mode of reproduction will be continuously ongoing with the production of new type of recombination due to the meiosis. So as and when they requires to change their character, that will be produced easily, that variation can be produced easily by this crossing over. So this is the significance story proving the process of evolution. So evolution is a result of this meiosis. So that's the another significance what we are getting from meiosis process, right? So please take the screenshot of this slide. Fine. Let me clarify the difference, main difference between meiosis and mitosis, right? Here, during prophase of mitosis and meiosis, you can see duplication of chromosome will contain two sister chromatid, whether here it is into the form of the tetrad. During metaphase, single mitotic plate, and here two metaphase plate has to happen, right? So single and double metaphase plate has to happen, and here two diploid cell will be produced, and here four haploid cells will be produced. So these are the main differences between meiosis and mitosis in the form of the process what we have seen till now. So please take the screenshot, and we are moving ahead with the last point of discussion. Each and every difference between meiosis and mitosis we are supposed to discuss now at the end of the chapter, right? So there are many events which are happened between mitosis as well and meiosis as well. So let us discuss each and every differences between both of them. Number one, occurrence, where it is occurring. Mitosis occurs in all the body cells including only normal cells, right? And meiosis happen only during into the germ cells, right? Into the germ cell only. This is into the body cell and this is into the germ cells. So gametogenesis, production of germ cell, is a result of the meiosis, and normal body cell is production of normal body cell is a result of mitosis. So this is the first change between meiosis and mitosis, first difference. Second, definition. It is equational division and reductional division. Equational, similar, same to same number, and reductional, it is creating haploid from diploid, so reduction is happening. Now number of daughter cells which are produced, 2 and 4, that's we know. That is mitosis produces 2 diploid cell and meiosis produce 4 haploid cells. That's what we have seen till now. Now difference between prophases of both. Mitosis, prophase, involves relatively few changes only, but in meiosis, prophase have distributed into 5 substages. Ready? Chromosomes or chromonema, not visible in prophase, but it is visible into the leptotin stage of prophase 1, chromomeres. Now synapses do not occur into mitosis. We know what is synapses. That is X shaped we can say pulling over of the hands of the chromosome on each other. It is the chromosomes which are getting overlapping to each other. It is synapses, which is not happening in mitosis. Why? Because there is no exchange of genetic material is seen, or it has to there into the mitosis, but it is occurring into the meiosis. So crossing over or synapses seen into the meiosis, not into mitosis. Crossing over as well do not occur, and it is occurring. Now metaphase. Difference between metaphase of mitosis and meiosis. During metaphase of mitosis, chromosome arrange along into the equator, into the one plane, where into meiosis, chromosomes arrange equally on either side of the equator, that is into two plate, into single plate. Ready? So that's the difference what we have observed. Now anaphase. Each centromere split into two cells. But in meiosis, what we know is centromere do not split into metaphase 1. It splits into metaphase 2, meiosis 2, but not in meiosis 1. So that's the difference. Now next difference, metaphase, that is centromeres orient toward equator while chromatid orient toward the pole, but centromeres orient towards pole while chromatid orient toward the equator at the metaphase 1. Now telophase. Telophase result into the formation of two daughter nuclei having the same number of the chromosome. That is why we have said it to the as the equational division. But during telophase 2, formation of two daughter nuclei which is having half of the number of the chromosome, that is of the parental cell. That is why we have said it into the reductional division. And cytokinesis followed immediately after karyokinesis and may or may not have occur at the end of the first karyokinesis. So that's the difference between meiosis and mitosis all over, and that's what you are supposed to remember. So main advantage what we are getting from mitosis is we can multiply and produce wear and tear business. We can maintain our body, we can produce many of these cells which are identical with genetic information, number of chromosome, etc. And by meiosis we can produce haploid gametes which are necessary for reproduction. So that's the importance of both meiosis and mitosis, and this is the difference between meiosis and mitosis all over. So that's with that discussion we are ending with the cell division and cell cycle. Ready? So I wish you best luck, and I wish you remember all the steps of the chapter, and we are moving ahead of the next chapter. Ready? So please take care, bye, and have a nice time.